Hello, my name is Mr. Chipman, and I teach biology, AP Biology at Murray High School in Murray, Kentucky. This is 5.1, meiosis. People say it both ways. I will probably say meiosis because that's how I was trained. I think it can be pronounced both ways. It literally doesn't matter. Let's talk about meiosis then. Meiosis is the process that creates gametes in sexually reproducing organisms. It is a process. It is not a cycle. We talked about the cell cycle in Unit 4. We talked about G1S, G2M, all those processes, right? And at the end of the cell cycle, you have a whole new cell, right, that does G1S, G2S, G1S, G2M, again and again and again. But that is not the case here. In meiosis, we have a process, and at the end we have a product, and that product is done, right, until some other process. And so at the end of meiosis, you have gamete cells. You have four genetically different gamete cells, and those gametes can combine with other gametes, and that process is called fertilization. That product is called a zygote, all those things that we're going to get into. But understanding those differences is going to be very important. We will talk more about that as the lecture moves on. And trust me, AP really sits on that difference quite a bit, asking you to know that. Let's talk about a couple things that are very important, some terms that we need to know here. One of those is haploid versus diploid. So humans are diploid organisms, right? We are diploid organisms because we get all of our chromosomes exist in a pair. So we have 23 pairs of chromosomes. Our mom donated half, 23, and dad donated the other half, 23. And so we have 23 pairs of chromosomes, half representing each parent. Those are diploid cells. Our normal cells are diploid cells, skin cells, muscle cells, whatever cells are diploid cells, each one of them having the full complement of DNA, and each one representing half of our parental genome, right? A haploid cell is where a cell has a single chromosome from each pair. I mean, if you had 23 pairs of shoes and you took only the left shoe, right, that'd be the example of that. And so those haploids are those individual pieces or those individual pairs of the chromosome, or half of each pair of the chromosome, what meiosis is making haploid cells, right? It is taking a diploid cell, a single diploid cell, and making four genetically different haploid cells from that. And not all cells do this process, right? Skin cells are not doing meiosis. Only reproductive cells are doing this. You have a reproductive cell, a diploid cell, and those diploid cells sort of serve as like the stem cells of the whole process, and they go through meiosis in order to create haploid gametes. Very important here. And there's another thing that we're going to talk about in the next slide. This is the idea of homologous pairs. We've already sort of mentioned this. So each chromosome exists in a pair, right? There's 23 pairs in the human genome. I'm using the human genome because it's something that is obviously near and dear to each of us, but dogs and guinea pigs and giraffes and oak trees all have pairs of chromosomes as well. They just have different pairs, different numbers of pairs. So I'm going to refer to 23 and 46 just because that's us, right? But just know that those numbers are not ubiquitous throughout all living things. So what is a homologous pair? A homologous pair represents one of those 23, or actually there's one that's not homologous. We'll get to that in this unit. But homologous is same, right? But not the same exactly. A great example, again, is a pair of shoes. So a pair of shoes exist together. They belong together, right? Your left and right shoes belong together, but they are not identical, right? They both belong to the same pair of shoes, but they are not identical shoes. So a homologous pair, so we have 23 pairs of chromosomes. Let's take pair number one. Pair number one, mom gave you one of those chromosomes, dad gave you the other one. Mom and dad do not have identical genes, right? Now, each one of those chromosomes have the same genes on them, meaning that they have those genes code for the same things. You're just getting different versions of those genes, and we're gonna look at those versions of genes in another lecture later, and this will all sort of come together. But it's sort of like I have blue eyes, right? My mom has blue eyes. My dad has brown eyes. How did that work? Well, my dad carries the blue eye gene, right? So he donated blue eyes to me. Mom donated blue eyes to me, and now I have blue eyes. So that exists on a homologous pair. A homologous pair, the two chromosomes are not identical, but they are similar to one another. They have the same genes. They just have different versions of those genes. This is very important. This concept of homologous pairs is extremely important. When a homologous pair replicates, this is when you have these pieces over here with sister chromatids, because sister chromatids is something different altogether than homologous pairs, right? You have purple and green. They're a homologous pair. Purple replicates, green replicates, and now you have 4. That brings us to interphase. Remember good old interphase G1S, G2. What happens in the S phase of interphase? DNA replicates, right? And so during interphase, you have this particular individual is a haploid 4. Speaking of, the prefix for, let's go back up here. I didn't mention these are very important. The abbreviation for diploid is 2N. Right, and so we would say that humans are 2N equals 46, right? Our diploid cells have 46 chromosomes or 23 pairs. And so N is equal to 23. So all of our haploid cells are going to have 23. Please don't make this more complicated than it needs to be. Don't. This is just a math problem, right? This is pre-algebra. Don't do it. So if this organism is 2N equals 4, right? Then when they replicate, this is still a diploid cell here after interphase. It's still a diploid cell. It's just that each chromosome has an identical pair, has a sister chromatid with it. It's not like we now have a pair of shoes that exist in 4. We just have 2 lefts and 2 rights. We don't have 4 different shoes. We have 2 identical lefts, 2 identical rights, right? And they go together. And so that's what you see here. One of the hardest things that students have is they want to make this a 4N cell, and that's not really it. It's a 2N cell that has twice the number of chromosomes. A 4N would be where you have 4 chromosomes. Each of them are like homologous, and that's a different thing. And so after interphase, the chromosomes duplicate. This is going to make sense because when we talk about meiosis, we're going to talk about 2 divisions, and you have to double the amount of chromosomes so that you can get to 23 at the end. I mean, if you just want to look at the sheer math of this, right? If 2N equals 4, then N equals 2, right? Each haploid cell is going to have 2 is what you see at the end. Well, how do you get from 4 and have 2 divisions to where you get to 2? Well, you have to multiply 4 times 2. You have to get 8, divide that in half, you get 4, divide that in half again, you get 2. So that's the idea. So let's look at the processes of meiosis 1 and meiosis 2. There are really just 3 total processes that I'm going to spend any amount of time on. And we will talk more about this in proceeding lectures as we get to 5.2 and we talk about crossing over. Yeah, but for now I'm going to mention these things, but just know that we're going to go into more detail in 5.2. During prophase 1, remember, during prophase, interphase has already occurred, right? So the DNA is replicated. During prophase 1, something happens that's called tetrad formation. So homologous pairs, these guys, they're already duplicated it during prophase. They're going to line up such that the purple and the green sort of stack on top of each other, right? Kind of like how my hands are currently lined up. My hands are homologous to one another, right? Are they identical to each other? Nope, they're not. They're not identical in that they're mirror images of each other. And then secondly, there's just little imperfections on both hands that make them not identical to each other, right? But they are homologous. They have the same parts. And so with this is sort of like when tetrads form, when homologous pairs line up during prophase 1, they do this. They literally lay against each other, and all the corresponding parts line up next to each other. Those corresponding parts
players and managers to report to the field house. Thank you.
So if you're a football player or a manager, you ought to report to the field house. So moving on. So they line up, right? And so if you switch the tips of my index finger, right, you'd still have tips of index finger on either one. It would just be a little bit different. And so these tetrads form. Tetrad means four. Literally, tetra means four. So instead of two pieces on each one, you have four, right? And so they line up. And then crossing over occurs. Again, we're going to talk more about crossing over in a separate lecture, 5.2. But crossing over is essentially where pieces of those homologous pairs can swap. This is actually good. This is like the index fingers swapping, right? Now, it wouldn't be good for my hand if you swapped tips of index fingers, but it's good for these chromosomes to switch places because this allows for genetic diversity. Genetic diversity is always good, almost always good. So that's the first step you need to know. Anaphase 1 is the second phase that you need to know. During anaphase 1, homologous pairs separate, right? Homologous pairs separate. Green separates from purple. Blue separates from red here, right? Notice that they've switched some red and blue to show the crossing over has happened, which I appreciate. So during anaphase 1, homologous pairs separate. Let's go back up to this picture. So one pair is blue, the next pair is red here, right? And so red and red are going to separate, and blue are going to separate. These new cells are haploid. Even though they have four pieces, they're haploid because they only have one of each pair. There's only one blue over here. That blue is duplicated, right? And there's only one blue here, one red. So these are haploid cells here. So anaphase 1, homologous pairs separate. Prophase 1, tetrads form, crossing over occurs. Metaphase, telophase, these things are very similar to what you see in mitosis, so we're not going to go over them. I would encourage you to make sure that you sort of re-up on those things. You might see them, but the real business end of meiosis 1 is prophase and anaphase 1. Moving on to meiosis 2. There is no interphase 2, by the way, very important to note. Right after telophase and cytokinesis, you have prophase 2. Prophase 2 is less exciting, so we're not even going to talk about it. Anaphase 2 is where we're going to talk about, spend most of our time. During anaphase 2, sister chromatids separate, right? So if you go back up to this picture, these two pieces are going to separate. These two pieces are going to separate, right? And so during meiosis 2, sister chromatids separate. And so at the end of this, at the end of anaphase 2 telekinesis or telophase cytokinesis, you have four genetically different cells, haploid cells, each one having half of the original complement, and they're genetically different because of a couple reasons that we're going to get into more in 5.2. Let's examine meiosis versus mitosis quickly. In meiosis, you have, well, first of all, what are some similarities? They use similar sorts of ideas and structures, sister chromatids, chromosomes, spindle fibers, all that stuff that makes cells are the same, right? What is different? Well, in meiosis, you have two divisions, right? You have meiosis 1, meiosis 2, and you end up with four cells, and those four cells are genetically different from one another because of crossing over and some things we're going to talk about in a later lecture. In mitosis, you end up with two identical diploid cells, right? They're called daughter cells. They're called daughter cells in meiosis as well, but they're two identical diploid cells. Meiosis is going to happen with gametes, or it's going to happen to produce gametes. Sometimes you'll see them called germ cells. It's less used. Mitosis is going to happen in somatic cells, which these are called body cells, like just skin cells, muscle cells, other kinds of cells. Let's talk a little bit about gamete formation here. Just some examples here that you might see. Oogenesis and spermatogenesis. Spermatogenesis is literally the creation of sperm. Oogenesis is the creation of an egg. Anytime you see this OO, that is egg. The difference between them is during oogenesis, you do have four cells that are made, but only one of them is going to be used. But it's a haploid cell. Whereas in spermatogenesis, you have start with 1, 2N, you end up with 4 haploids like you would expect to see. And then fertilization is N plus N equals 2N, and that it has now mom and dad's genes. So that's what you see here, zygote. Fertilization equals sperm plus egg equals zygote. Zygote's a diploid thing all the time. Gametes are haploid. Good time here. Here's a karyotype. A karyotype is a picture of chromosomes. We're going to get more into this as we move throughout this unit. We will look at karyotypes, and there's a karyotype there, which looks like some sort of like one of those pictures. I forget the name of them. Rorschach, maybe? Let's look at a practice problem. Which of the following best explains why meiosis is necessary in sexually reproducing organisms? Sexually reproducing organisms produce gametes. Meiosis allows for the production of 4 genetically different gametes. Let's look at ABCD and see where this is taking us. It ensures that gamete contain half the number of chromosomes, so the diploid number is restored after fertilization. Okay, yeah. N plus N equals 2N, right? We like that one. Let's look at the other ones, though. A's got me a little unsure. Produces identical daughter cells. Doesn't. Allows for the growth and repair of creating new cells. Nope. Because that's not the purpose. Replaces old somatic cells with a nope. A is the correct answer because B, C, D are obviously wrong. I hope this was helpful. Meiosis is fairly difficult, right? It's a big front end load, but as we move throughout the rest of this unit, it'll start to sort of come clear as we talk about alleles and dominant and recessive and all that business. Thanks for watching.